Okay friends, welcome to this last session of the day, and I do like to join thanking both Steve and Tom inviting me to this wonderful symposium honoring Govind. And Govind, greetings to you from Bangalore, and since I'm the only one coming from India, I take this opportunity to greet you on behalf of the rest of the fraternity also. Govind has been a source of information, a source of inspiration to all of us, and Govind commands a lot of respect from all of us. And I remember last time Govind was there in India was, I guess, in 1983. That was the genetics conference, and the hall was really overflowing. I think they knew that they had to make a big hall for Govind, but that was not big enough. And the people were going all over the corridor and still trying to get a glimpse of what Govind was going to say. So with that, let me then say that we have 4 speakers in the session, and the first one is John Hwa. And the abstract of his talk will be on page 56 and 57. John is at Dartmouth Medical School and Dartmouth Hitchcock Medical Center in Hanover. And John has been with Govind, I guess, around 1960. Yeah. And so John, you have 18 minutes. But I get up. And then I start doing dance here.
I'm waiting for the dance. I'd first like to thank Tom and SNiP for organising this wonderful symposium. I was with Govind from 1996 to 2000, and it was a wonderful period of my life, and I've learned an incredible amount from Govind. He's been a wonderful mentor and has had a great influence on the development of my lab as well. So today I'd like to describe some of our recent work on the pharmacogenetics of the human prostacyclin receptor, and thus the title HIP, SNiP, COX, and Vioxx. HIP is the International Union of Pharmacology nomenclature for the human prostacyclin receptor. SNiP is single nucleotide polymorphism. COX is actually not this gentleman at the back of the boat here, but the enzyme cyclooxygenase. And Vioxx is a selective COX-2 inhibitor that was recently withdrawn because of cardiovascular side effects. And so by the end of the talk, I hope to demonstrate there's an important relationship between HIP, SNPs, and COX and Vioxx. But just before I start, this is a picture of the lab back in June of 1996. And here's a picture of Govind with his trusty camera. Tom is here as well. There's a whole host of characters, many of them are in the audience today. And this is me when I still had black, dark hair. And actually these days I'm looking a little bit more like Harold. So I've been studying the human prostacyclin receptor since I left Govind's lab. It's a typical class A 7-transmembrane G-protein coupled receptor. And in the cardiovascular system, it is expressed predominantly on platelets, where it prevents platelets from aggregating and clotting, and vascular smooth muscle cells, where it promotes vasodilatation. And since its discovery in the 1970s by Sir John Vane, it was felt that it's somewhat the poor relation to nitric oxide, in terms of many people thought that it had redundant activities, and in its absence, nitric oxide could easily compensate for its function. However, in the late 1990s and early 2000s, the prostacyclin receptor was knocked out from mice. And these demonstrated and started giving a clue that maybe these receptors and the signaling of prostacyclin itself was important in terms of these mice had enhanced thrombotic effects, they had the vascular proliferation, and showed evidence in the female mice of pre-menopausal atherogenesis. But one of the major questions remain: is it important in humans? And is it a redundant factor? So this is taking you all back to Biochemistry 101. Phospholipids via phospholipase A2 arachidonic acid. There's COX-2 predominantly in the endothelium in the cardiovascular system, COX-1 in platelets, COX-2 leading to the production of prostacyclin, and COX-1 leading to the production of thromboxane A2. And back 30 years ago, John Hwa postulated that there was a dynamic balance between thromboxane and prostacyclin, where in terms of the cardiovascular system, thromboxane was probably bad and prostacyclin was probably good. And so there was indirect evidence from drug trials on humans that prostacyclin may be important because low-dose aspirin, which is a selective COX-1 inhibitor, reduced thromboxane and removed the bad, and so we know that low-dose aspirin is predicted against heart attacks and strokes. And then more recently with Vioxx and other selective COX-2 inhibitors, it's believed that they inhibit COX-2 in the endothelium. And via this, many now believe that it reduces the production of prostacyclin and prostacyclin signaling. And as a result, there is increased thrombosis, vasoconstriction, and the cardiovascular side effects. And so there's a number of indirect evidence that pointed towards prostacyclin playing an important role, possibly in humans, in cardiac protection. So there are the selective COX-2 inhibitors, which selectively inhibited COX-2. And then there's the mouse experiments where the prostacyclin receptor was knocked out. So we came up with a simple hypothesis. If we could find a human knockout, then we could look for the importance of prostacyclin in humans. And of course, I mean, we couldn't do it genetically, and so we thought a simple way of going about it was if we could find a dysfunctional SNP, a polymorphism, a genetic variant, which knocked out the function of the receptor, then essentially we would have a functional human knockout. And from this, we could examine the importance of the prostacyclin receptor in the actual human tissues and subsequent clinical sequelae. And so this is the prostacyclin receptor with its 7 transmembrane domains. There's disulfide bonds on the extracellular side, areas of glycosylation, looped on the outside, looped on the inside, palmetylation on the C-terminal tail, and a very, very long C-terminal tail. And so we set up a study which we call the POP trial. Pharmacogenetics of prostacyclin is the all-important IRB, Institutional Review Board number. And initially, it actually started off as a collaboration between 6 different departments at Dartmouth. And progressively, over the more recent years, we've actually had a number of other centers and PIs approach us. And we now have a collaboration of 12 national and international centers. And we have approximately access to just a bit over 22,000 genomic samples and information on patients. So the phase 1 of the trial was pretty simple. What we did was we obtained blood or tissue samples from patients, isolated genomic DNA, PCR amplified it, and it's essentially just sequence. And then if we did find from our clinical database of patients, if we did find a disease association, then we search for mechanism. What is the pathophysiology? Why is there, for example, increased cardiovascular disease in these patients? What is the signaling mechanism? What is going wrong with signaling? And finally, down to the fundamental structure function. What is the structural defect that is associated with these individual mutations? And there were 2 MD PhD students that spearheaded this, Eric Earhart and Jeremiah Siphon, and we really tried to run all the way from fundamental basic structure function all the way to the clinical arena. But just before I progress any further, this was back in 2000 when the lab climbed Mount Cardigan, and we were all very exhilarated after a long climb getting to the top of the mountain and having this picture taken. But we could always trust Govan to bring us back to Earth. This whole board is full of hype. A little room for science, please. Govan was a very, very tough mentor, and he made sure that science was our #1 priority. And so he left many of these notes around the lab, and I was fortunate to have kept one of them. So now to the science. So far in phase 1, we have sequenced the coding region of about 2,500 patients. And from these 2,500 patients, we have found 39 distinct genetic variants on the prostacyclin receptor. 23 of them changes the amino acid, okay? And 16 of them does not change the amino acid. There's synonymous mutations. And you can see they're distributed throughout the transmembrane domains. There's a number in the third intracellular loop, and there's a number in the C terminus. Now, if you took 100 Asians, 100 Caucasians, 100 African Americans, 100 Mexicans, and we went and sequenced the prostacyclin gene in those. These are the frequency, and this serves to demonstrate that there are two very common mutations, okay, and then the other mutations are very distinctly distributed between the different racial groups. And then after this, we went on to characterize them both from the binding perspective, the activation perspective, and cell surface expression. And without going through all the data, I'd just like to highlight one of the mutations, and that's the R212C, which is found in the third intracellular loop. It's 50% conserved across all prostanoid receptors. It has normal binding, abnormal activation, and abnormal cell surface expression. And so here's a three-dimensional molecular model, which was composed by Scott Klein, who's in the audience today. And this R212C is located in an alpha helical region in the third intracellular loop, and it's believed to interact with a serine at position 205. And we've gone through and progressively mutated both the serine 205 and did perform random mutagenesis on the R212C to demonstrate that there was this functional defect associated with it. And this is just to show that we have sequenced a number of patients, and this was a sequence of the wild type, of a heterozygote, and of a homozygote patient. So this was a random mutagenesis training the threonine, the cysteine, which is the naturally occurring mutation. Interestingly, in an Asian population, there's also a histidine that can also be a naturally occurring mutation at this point. Proline, serine, and leucine, and you can see that all these numbers are different from the wild type, demonstrating that there is a defect associated with this receptor, even in a COS1 cell overexpression system. So the next question is, this receptor is found predominantly on platelets and on vascular smooth muscle cells, and what do the patient's tissues actually show? And so in terms of platelets, we know that the prostacyclin receptor inhibits this process of clotting. And from wild type and R212C, we looked at the amount of cyclic AMP that they were able to produce, and you could see that the R212C was significantly deficient in function as compared to the wild type, which is shown here in white. And this is performed at a number of different concentrations of an agonist, iloprost. Then, if you have a look at function in terms of the ability to aggregate the platelets, wild type prostacyclin receptor, when activated, can stimulate a robust amount of cyclic AMP, which can actually inhibit these platelets from clotting, as opposed to the R212C, which is right down here, in which they couldn't produce much cyclic AMP, and they couldn't prevent the clotting process from happening. And so, in the R212C patients, they have a relative state of hyperthrombosis, and so they have a greater propensity to form blood clots. And this can be reflected by some of the clinical data in which a blood clot is the sort of the final common pathway for both a heart attack and stroke. And so if you take the R212C patients and age risk factor match control patients and look at the number of events, heart attacks, strokes, blockages of blood vessels in the leg, the R212C patients have many more of these events as compared to the control. And then another way of looking at it using a Wilcoxon rank sum test, you can look at the number of events over a 4-year period for the wild type versus R212C. And the R212C consistently had more cardiovascular events as compared to the wild type. And this, in many ways, reflected what was seen with Vioxx. In many of the patients had more cardiovascular events as compared to risk factor match controls. So the next tissue is vascular smooth muscle cell, and this was in collaboration with my wife, who's in the Department of Vascular Surgery, and it's fortunate that we were able to get actual human smooth muscle cell samples from bypass operations. And this is what a contractile quiescent healthy vascular smooth muscle cells look like. And if you stimulate it with a lot of the different cytokines, for example, you can get to this synthetic proliferative phenotype, and it's this synthetic proliferative phenotype that ultimately leads to the thickening of the blood vessel, the ulceration, and the associated heart attack. And Kathy, along with a number of her colleagues and ourselves, demonstrated that the prostacyclin receptor actually inhibited this process, and so maintained it in a contractile quiescent state. I haven't got time to go into the details of a lot of these experiments, but essentially she showed that the prostacyclin receptor via pKa induced this quiescent differentiated state. It causes induction of more prostacyclin, which allows sort of a cascade effect of prostacyclin, maintaining the thickness of the blood vessel in a healthy state. And it's via a number of different pathways, the ERK pathway and inhibition of AKP1. So this vascular smooth muscle cell proliferation reflects the severity of disease. And so in all these patients that we examined, they've had coronary angiograms in which dye is squirted into the blood vessels around the heart, and you can see the amount of narrowing that's in the blood vessels around the heart. And if you have a look at the R212C patients versus the cardiology patient, there are three main blood vessels that supply the heart. And in these risk factor match controls, the R212C patients have more severe disease as compared to the cardiology controlled patients. And this is just another way of looking at it, in which the R212C patients have more three-vessel disease as compared to the cardiology control patients. And so this is what actually a normal angiogram should look like, in which the blood vessel is outlined by very clear tubes. And this is what our homozygote patient looked like, in which here's the dye being squirted into the blood vessels. This patient has very severe disease because he's actually blocked off two of the major three blood vessels that supplies the heart. He's had a bypass operation, it's actually blocked off his bypass operation. And he's essentially surviving on a rather tatty looking single left circumflex blood vessel. We were fortunate at the time that we were doing these studies that we were approached by a group, an Italian group, who had a population of about 60 patients with what's called deep vein thrombosis, in which they've clotted off the veins in the legs. And they were interested to see whether possibly the prostacyclin polymorphisms or genetic variants could account for some of the problems with their patients. The conclusion from their study was no, it didn't actually, wasn't actually associated with the DVT. But what was very interesting was all their patients had what was called carotid ultrasound, in which they had a look at the blood vessels in the neck. And this is what a normal intermedia thickness looks like. You can see this level, thin intermedia outlined here. And this is what an abnormal one looks like, in which it's irregularly thickened and very abnormal. And this is the beginning of an atherosclerotic plaque. And what they found in the R212C patients was unfortunately they only had 4 patients out of these 60, but each one of them, not so much this one, but these 3, had much, much thicker blood vessels as compared to controls. And so this demonstrated and helped support our notion that the R212C increases the vascular disease in these patients. And so I've described one of our mutants which has a functional defect and associated with cardiovascular disease. And we're in the process of studying many more of the others and systematically looking at them in our population and in a number of other populations as well. So just to quickly summarize the POP trial, there's 39, we found 39 genetic variants from sequencing about 2,500 genomic samples. We characterized these mutations, and many of them has provided some important insights into the human prostacyclin receptor. The R212C is functionally defective in platelets, and as a consequence of that, they have increased cardiovascular events. It's functionally defective in vascular smooth muscle cells. As a consequence of that, there's increased burden of cardiovascular disease. And so the clinical implications of this is that, towards this concept of personalized medicine, in the future, when everyone's genome is sequenced, we may be able to search for these patients with R212C and try to control their risk factors early and maybe start them on a low-dose aspirin earlier. So this was a picture of the group when I left the lab. And Christoph, and then later myself, wrote a poem when we left the lab, down to the room of artificial lights. I lose track of the days and track of the nights, describing the long hours we put into the redox work. And then finally I concluded with science and 7 hours of friendship I cherish, the memories of good times will never perish. This is a recent picture of my lab at Dartmouth Medical School. These are the collaborators that we've had over the years in this HOP study. This is just to indicate some of our international collaborators, and we're hoping over the next few years to expand this even further and get many more tens of thousands of patients. Acknowledgement for kind support of NIH, American Heart Association. And for my friends and colleagues, we're actually moving next week to Madison. That's Madison, Connecticut. And we'll be going 3 hours down the road, and the labs will be moving to New Haven. And then just to finish, a confession. Govan came back from Japan in 1998, and he left this message in the tea room. People, not a very nice sight to come back to. And that was because the tea room was an absolute mess. And so I have to confess that I partially contributed to it. But the reason for it is not because we were partying in the tea room after hours. It was because everyone was spending long hours working in the lab after hours, even in the absence